DNA is a long molecule that contains our unique genetic code. Like a recipe book, it holds the instructions for making all the proteins in our bodies. It's what decides if we have green or blue eyes. DNA is found in the nucleus of the cell. DNA molecules are tightly packed around proteins called histones to make structures called chromosomes. Human beings have 23 pairs of chromosomes in every cell, which makes 46 chromosomes in total. Prokaryotes have one chromosome, consisting of a circular DNA molecule. Some prokaryotes also have plasmids, but eukaryotes do not. Eukaryotic chromosomes are much bigger than those of prokaryotes. In eukaryotic cells, replication may begin at dozens or even hundreds of places on the DNA molecule, proceeding in both directions until each chromosome is completely copied. DNA contains 4 basic building bases: thymine, adenine, guanine, cytosine, and an RNA uracil.
Transcription refers to the process when information in a DNA strand is copied to make a new molecule of mRNA. Transcription occurs in the cytoplasm or nucleus and is aided by the enzyme RNA polymerase.
RNA polymerase attaches to the DNA at the promoter. It then unwinds and unlinks the 2 strands of DNA and uses one of the DNA strands as a template. It then matches nucleotides with this complementary pair and binds a new RNA to form a copy of DNA. DNA can be changed, resulting in different types of mutations. A substitution is a mutation that exchanges one base for another. For example, a change in a single chemical letter, such as switching an A to a G. Deletions are mutations in which a section of DNA is lost or deleted. Insertions are mutations in which extra base pairs are inserted into a new place in the DNA. There's also external forces that can affect the DNA, such as chemical things. Spliceosomes occur in the nucleus. Their function is to process pre-mRNA by splicing out intronic nucleic acids. They also produce mRNA, which is then translated to protein in ribosomes. The spliceosomes, also called SNRPs, have to recognize the intron and extron boundaries via the splice sequences and multiple over weak signals. They also catalyze and cut and paste reactions that remove introns and join extrons. Once the DNA is turned to RNA, it goes into the cytoplasm. From the cytoplasm to the ribosome and ribosome to the RER. Translation is decoding of an mRNA to make a polypeptide. There's a codon of mRNA that reads 5 to 3 by the tRNA. The tRNA has an anticodon which pairs with the codon of mRNA through base pairing. The other end of tRNA carries the amino acids. The pairing of tRNA and mRNA pair inside the ribosome. The end result is a peptide chain which mirrors the sequence of codons in the mRNA, making a protein.
There are 3 processes to translation. First, there's initiation. In this stage, the ribosome gets together with the mRNA and the first tRNA, so translation can begin. Second, there's elongation. In this stage, amino acids are brought to the ribosome by tRNAs and linked together to form a chain. Lastly, there's termination. So this is the last stage. The finished polypeptide is released to go and do its job in the cell.
Ribosomes consist of a large subunit and a small subunit. When these 2 subunits combine, they form a complete ribosome that converts genetic code found in RNA into a sequence of amino acids. The ending result is a protein structure. Complex structures, which physically move along the mRNA molecule, catalyze the assembly of amino acids into protein chains. They also bind tRNAs and various accessory molecules necessary for protein synthesis.